Next, I want to talk to you about the pattern of inheritance that might again deviate from what your expectations are from the straightforward dominant recessive Mendelian genetics. And that is this idea of epistasis, which is where one gene alters the expression of a gene at another locus. And so in order to figure out what the phenotypes look like, you actually need information about the two different genes. In this example that you see on your screen, we have, for example, looking at coat color, which is a gene. The mice can either be black or brown, but there's a completely different gene that determines if that coat color actually shows or not. And so we need information about both of these different genes to figure out what the coat color is going to look. And that's why you actually see three potential phenotypes on here. Some of them are going to be white because they might have the allele combination that says that we don't show any coat color, regardless of the combination of alleles for the black and brown. So let's take a look at some questions that deal with this idea of epistasis. Okay, so let's scroll down. In a species of cactus, a gene controls the length of spines. So we're going to do big L is going to be long spines, and that's going to be dominant over little l, which is short spines. A second gene determines whether the cactus will produce spines or not. Homozygous recessive little s individuals have no spines. These genes are unlinked. So what is the relationship between these two genes? All right, we've just talked about this. We know that this is epistasis. All right, when we look at a cross between a true breeding short-spined cactus, you can see the genotype here, and a spineless cactus, you can see part of the genotype here, could never produce, and our sheet got a little wonky here, there we go, could never produce a spineless offspring, all long-spined offspring, all short-spined offspring, or a combination of long and short-spined offspring. Now the correct answer here is that it could never produce spineless offspring. And that is because we are always going to have that condition of having basically the spines shown, because we have that big S in one of the parents, or that homozygous dominant condition in one of the parents, okay? Let's take a look at the next section here. So, if two doubly heterozygous cacti, you can see the genotypes there, were crossed, what would the F1 phenotypic probabilities and ratios be? Let's take a look at the Punnett squares. We can do two separate basically ratios here to take a look at what would be the probabilities. So in terms of the gene controlling the spine expression, whether you show spines or not, we can see that we have a big S and a little s, with a big S and a little s individual coming together. And so we would have, I'm going to make my little s's look little, just so that they're easier to see. Okay, so we're going to have 3/4, you can see right here, 3/4 are going to have spines, and 1/4 is going to have no spines. Now let's look at the Punnett square for either long or short spines. So we have, let's see, a big L individual and little l with a big L, little l. And let's see, let's move this down so we can, so it doesn't get deleted. So we have a big L, little l, big L, little l. Okay, And so what we see here is that we're actually going to end up with 3/4 that are going to have those long spines and 1/4 that are going to have short spines. Now we can look at the phenotype probabilities and ratios. And we have to combine the information that we got from both of our Punnett squares, from here and from here, okay? So we can now look, I'm just going to write this down here again so that it's easy for us to see. So 3/4 are going to have spine, well, 3/4 have spines and 1/4 have no spine, okay? And so now we can figure out that we can have individuals who are long with spines, okay? And that's going to be 3/4 times 3/4 or 9/16. We're going to have short spines. So short with spines is going to be 1/4 times 3/4. Okay, and that's going to be 3/16. Next, we're going to have no spines, and individuals with no spines are going to be both those who have long and short, but who have the no spine combination of alleles for that other gene, okay? So no spines basically can be, if we look at no spines long, with long spines, we would see 3/16 total. So 3/4 times 1/4, 3/16. And no spine and short would be 3/4 times the same, that the short, which is 1/4. Okay? Sorry, I made a mistake here. This is no spine, which is 1/4, times the short, which is 1/4, and so that's going to be 1/16. But these two combined are the phenotypes for no spine. So it's really going to be 4/16 if you add these two. Let's see. If you add these two numbers up, you're going to get 4/16, which are going to have no spine because they have this combination that regardless of having short or long in the other gene, they're just never going to show spines.